And now we remain, let's say, with the Basque speakers, even though Justin is not, let's say, the most representative person of the Basque people. But so Justin Barry from the pharma company Mediatek company will present, let's say, another experience with another pilot line called Nanofacturing Project, which has been funded under the same call as Iridice One. And so Justin Barry will also share his expertise from a slightly different perspective, because Mediatek is developing a product, so this is a different perspective. So Justin, the floor is yours.
Thank you very much, Patrick. Good morning, everybody, or nearly good afternoon. I will try and keep on time and make sure you get to your lunch. Patrick said you're probably getting hungry. I can understand that. So on the program, that's what it says, the pilot project. So I don't want to disappoint you. I will give an update on where we are with that. What Patrick actually asked me to present is slightly different. So I don't quite know where the overlap happened, but anyway, from the bench to the clinic via a robust quality system. You can see a pattern developing over the last 3 lectures about quality systems. The quality system is the way in which we create the environment for the production of the product that shows that it's the correct quality, efficacy, and safe. But first, before I've said I will, before the program, because the program said I will, I'll give you a very quick update on nanofacturing. The update we gave to the project supervisor about 2 weeks ago was 85 slides. I've tried to cut it down a little bit. The objective is the development of a medium and large-scale sustainable manufacturing platform for clinically compliant solid core nanopharmaceuticals. Iraida is doing polymeric. I think it's a reasonable description. That's one class of nanoparticles. Mediatek, we deal with solid core. They are what we're calling ultra-smalls. They are a gold core-based nanoparticle with a corona, which we can functionalize. The core is around 1.6 to 1.8 nanometers, so we're right down at the other scale. So the project has multiple objectives, as all these projects do, but I think I want to kick out the 2 really important ones here. The objectives, in the same way that Iraida's group is looking at an open access plant or a plant that could be useful for people developing those kinds of nanoparticles, we are trying to do one that will deal with solid core nanoparticles and give an open access plant in Europe for the process development and scale up of those nanoparticles. It's with one of our consortium partners, CPI in the UK. That is a very, or at least I believe it's a very, very important objective. If we're successful at that, we will at least achieve something that can be useful across the European field in nanoparticle development. And we also have this one, a full spectrum of robust and practical chemical and biological testing. Now it's interesting, very, very quickly, when we started this project in February, 2015, the European NCL was just kicking off. It wasn't really up to speed, and I'm hearing from Simon and everybody else that it's really running very fast. So we are going to spend some effort in getting in line with those guys, because we need a common approach. I will mention that when we did the first clinical trial with our nanoparticles in 2012, we did that in Switzerland. We had 22 questions back when we put in the application. We managed to answer them all reasonably satisfactorily, and they came back with another 8. So we answered those as well, and we got permission to do the trial. And when we applied to the FDA, not sure whether Neil was responsible for that, we got another 11 very deep, far-reaching questions about nanoparticles. We did answer them. We didn't do the trial in the States in the end. We did it in Australia because it was cheaper. But the point I'm getting at is that there still seems to be a hell of a lot of work to be done on what is a robust and practical chemical and biological characterization set that will give some reasonable indication of safety to the regulatory authorities. Very quickly, work package 2, selection of key ligands is functionalized. This is just where we are. The ligands, we have functionalized ligands, or ligands to functionalize the nanoparticle. We had to create a set that we thought were of key importance. It's done. The synthetic routes of those from the bench to a GMP environment is a fundamental step, so we're working on that, and some of those routes have been elucidated and in place. The construction of a new non-sterile facility at our place in Bilbao is cracking on quite well, and we have yet to crack on with the second part of that, which is the aseptic suite, which is also work in progress. The scale-up, we've got up to 5 and 10 liter scales now. We've based all our current production on 2 liter scale reactors, but we have to move up from that, so 5 and 10. And then CPI are working on continuous flow systems and being, I must say, extremely successful at it. So we're very, very optimistic that we're going to hit all those goals and come up with a means of manufacture at industrial scale and GMP compliant. From the characterization particles, the W6, work package 6, there's a new paper being published on ultra-smalls, which has some interesting key issues concerning non-specific protein binding, which everybody knows is one of the fundamental pieces or one of the fundamental concerns of systemic circulation for nanoparticles, and we have some interesting observations there. We've got new characterization tools that we think are useful, but as I said, we're going to be starting talking far more closely, I think, with Simon and the NCL in the EU going forward. WP7 is not relevant right now, and WP8, lots of stuff there, and I'm even going to put up some brochures if anybody's interested at some point during the day. The inspector said we were impressive at the 18-month review period. I'm not quite sure what impressive means. He might have been referring to the number of slides, or he may have been referring to the details, but we'll see when he gets his written report. That's just a quick shot, because a picture paints 1,000 words, of where we are with the new non-sterile manufacturing suite, and that kind of links in to the next part. So I want to go back to GMP. One more comment on that. Ireda said, you know, the TRLs, that's great. For my sins, I'm part of the board of the IMP QP working group in the European QP Association. And we spend amazing amount of our time trying to figure out if there is any difference in the rules and regulations between investigational medicinal products and full-fledged pharmaceutical regularly produced products. And the conclusion we're forced to is the answer is no. Bottom line is that if you go into a clinical trial, we're going to be putting these things into human beings. So the idea that you can kind of step back and say, well, it's okay because they're investigational, so we don't have to worry quite so much about it, that doesn't fly anymore, I'm afraid. And we find, as the regulations are revised year on year on year, and they are, every single year there are modifications, the dividing line between IMPs and fully fledged products is disappearing. That sounds like a threat. It's not really a threat. It's simply a position that really defines the reality. We're putting nanoparticles into people. We have to be very careful about what we do and how we do it, and very careful about what we know, and proving that we're not going to cause any harm. That's the other thing that we're seeing today as well, risk-based. It's not only a great idea, it's not only a good tool, it's actually obligatory. The FDA beat Europe to that. They said quite recently, you will base your quality system on a risk assessment. That's not an option, that's not a, we think it's a great idea, you might consider it. That is, you will do that. And the Europeans have done exactly the same. So we are now expected to have a quality system that is based on risk. It makes a lot of sense. The system is designed, obviously, to ensure that the product is of the correct quality, that it is efficacious, and above all, that it's safe. With that said, the first license we got in Midotech was in 2012. There were 11 of us. That's not very many people. It was a full process from start to finish. It can be done with relatively little resource. There's a lot of sort of angst about that. People say, gosh, it takes a lot of work. We really haven't got enough people. If you go at this in a methodical way, it can be done. We've done it, and lots of other people have done it. It's not a nightmare, but it is a lot of work. It's still based, at least my feeling is it's still fundamentally based around the plant you're doing the work in, the processes you actually got in place, the product you're producing, the procedures you're using to produce it, and the people who are doing the work. It's pretty straightforward and common sense. That's a selection of all the junk we're faced with. It's an absolute mess, but it is nevertheless all relevant, and there's a lot of reading there, I'm afraid. But it comes down to how you go about proving that your product is safe. So we look at the quality system from the point of view of the premises, the process, and the personnel. We have The process on one hand, which is all about the suppliers, the raw materials you're using, the procedure for manufacturing, the analytics you use, the procedures, the validation, tech transfer, all that lot. You have work around the premises, the maintenance, cleaning, validation, cleaning, plant maintenance facilities, and so on and so forth. I'm not going to read every one of these. The personnel, the qualifications, the experience of the personnel. Are they up to scratch? Have you trained them to do what they're expected to do? That gives you your product at the end of it, and then you have the final piece here, which is quality control, QA release, and QP release. One of the things I took out in the last couple of days when Patrick reminded me that I had not very many minutes to do this was the slide on why quality has changed over the last 20, 30 years. It used to be a very, very product test-based system. You tested the product at the end of it and showed that it was of the right quality. Nowadays, we don't do that so much as prove that the process from start to finish is inherently designed to give you exactly what you want. So we look at the quality system from that point of view. Again, the process, risk analysis, the raw materials, the actives, the critical process parameters, all these acronyms come up, the CCPs, the CPPs, the key quality attributes, these things Iraida also mentioned and commented on very thoroughly, so I won't repeat it all. But that defines, or those define, how you control your process, and it tells you what you need to test. And believe me, if the point at where you are in your process, you don't have the tests in place, then you haven't got a product anyway. So that is a fundamental step about your process to get to where you need to be in order to do the right testing to show that the right attributes and the right quality attributes have been met. The quality system premises then, this is about the environment. This is about where you're doing it, how you're doing it, what you're doing in. People know about Class C, Class B, Class A rooms, an isolator. Is it an aseptic process? Is it a non-aseptic process? So what equipment and what instruments are you using? Are they installed and documented correctly? Are they validated to the degree that is necessary? There is some gray space there with the validation, although if you talk to the German regulatory authorities, they're a lot less flexible than some others. I had one instance where a regulator, a German inspector, stood up and said, you will validate fully all your analytical procedures for a phase 1 clinical trial. We had a spectacular argument about that. I'm not sure still who won, but we haven't applied to BfArM yet. But be warned, it is to some degree about interpretation from the regulatory authorities as well. What controls are you using for your working area? Is the area segregated from other activities? That's really evil in terms of an R&D environment because so many different activities can be done in the same place. You really have to be careful about keeping a nice, clear segregation between products. Are the personnel and activities properly segregated? That's repeating myself, sorry. So from a personnel point of view, very quickly, documentation, organization, training records, you can read all this, I'm not going to go through it, but it is a fundamental piece of the quality system to prove and show, and it's actually only fair, that the operators doing the work, or the person doing the work, is fully trained and knows what they're doing. It sounds straightforward, but quite often you can walk into a place when you're doing an inspection and ask somebody, and they don't actually really understand what they're doing overall. They have a micro piece of the process, and that's not effective. If you look at the quality is only as good as the people making the product, that's a fundamental axiom, then clearly the people are really, really important in the process. It does create a lot of paperwork. That's just one product. We've got 4 of these, but there's 101 SOPs, 53 worksheets, That's a lot of paper, I'm afraid. But again, if you take a rational, logical approach, it doesn't have to be a nightmare. So if you build the process, or you build the quality system, fundamentally starting with the process, map the process, and look at the risk assessments of the critical points, which parts of the process are producing the changes or providing the attributes that you want in your product. Those are the bits where you have to pay attention. Those are the bits where you'll put your control testing in to show that they don't vary. Again, this is pretty basic. That gives you your critical process parameters, the critical quality attributes, and the key performance indicators. And there's lots of other acronyms, but it boils down to the same thing. What changes where? How do you stop it changing if you don't want to change it? Or if you do want something to change, how do you control that change in the process to provide the next step in the synthetic route or the next step in the synthesis of your nanoparticle and show that it's always the same. So building the process outwards defines the process and specification, defining your raw materials and their control, define the testing you're performing and the methods, set the limits, define the acceptable quantity, and that gives you the famous document, which is the product specification. The product specification is the bit that tells you what your product is, and it tells you how you've proved that it is what it is, and how you continue to prove that it is what it is, and it doesn't change from batch A to B to C to D to E. I think Simon in Biospain a couple of weeks ago or last week mentioned that he sees lots of applications where product batch 1, batch 2, batch 3, and batch 4, the data presented, prove quite conclusively that there are 4 different products. That wasn't the aim. The aim was to have 4 batches of the same product. That beastie is the one that will help you tell you that you've got that. And that's critical because if we want to scale up, once you have the specification, you can show that if your process is scalable from a chemistry point of view, that you've been successful. It will tell you if you got it right. It will also, by the way, tell you if you didn't get it right. But without the product specification, if you think about it from a fundamental basic development point of view, the quicker you get something in place, the easier it is to control what happens when you scale up. And we're all talking about scaling up. We're all talking about making lots and lots of nanoparticle preparations. We'll never get there if you're not very clear on how you prove that the 100 kilo batch you did is the same as the 1 gram batch you did several years ago or several months ago in your lab. Patrick, you've just got up and I'm pretty close to the end. Yeah, I did have to do 2 presentations in one, be fair. Okay, so what you do will define your quality policy. Describing the process gives you the product and the specification. Describing your personnel defines the organization. And where you do it and what equipment you use gives you your plant and procedures. So we're back to these key words here. Just a word about analysis. I haven't mentioned it. I'll do it very quickly. As to where analysis fits into the pharmaceutical quality system, the answer is everywhere, which is why we're getting also hectic about analytical techniques and procedures. There are lots out there, and nanoparticles raise very distinct challenges. That's a comment for another time. I will, last slide, promise, okay? Just a comment about the regulatory authorities, and again, because historically our attitude has changed quite rightly. When I started this life in the Wellcome Foundation and then actually GSK, the attitude was don't talk to the regulatory authorities, they're a nasty bunch of people, they'll tell you what you can't do and try and stop everything you do, which is patently ridiculous, but that was a mindset in those days. They were seen as a barrier. They were seen as unapproachable. They were seen as dogmatic and regulationally obsessed. I know for a fact that Neil certainly doesn't fit that profile for a kickoff. They really are, and I've spoken to the MHRA, the FDA, the BfArM, apart from that guy with the small validation issue, Swissmedic, the Spanish regulations, and various other groups and things. They are keen on new technology. They want it to work. They are very willing to listen, and they are looking for solutions and trying to help us get there. They are desperate to facilitate new treatments to patients. That's what they're for, except that that one still applies. It will never fly if it isn't of a quality that is appropriate and if it's safe and is efficacious. Get that lot right, talk to the authorities, work with them and work through it, and you're on a good path to success. It's always a team. Thanks very much indeed for your time.
Thank you. Thank you, Justin. Great talk. So we have time just for a couple of questions, 2 or 3 minutes, from this great ground expertise developed by Justin and his team. Any question? Or maybe most of the participants didn't reach, this stage in the development of the product. So there will be, let's say, future clients to MediaTek maybe in a couple of months, I hope. So,
a lot of hunger in the room.
So, oh yes, definitely yes. So, if there is no question, so I will close the session. I would like to thank you very much for having participating to this last session on QBD. I hope it has opened your mind and your spirit. And now you have seen, let's say, some key people, key experts in this area. So please feel free to contact them during the lunch or during the rest of the assembly. And is Alex in the room? Do we have special instructions? Oh,
Hi,
sorry.
Hi, Patrick.
So
I'm always behind you, you know.